Cellular Interactions, Part 1. From the beginning of the course, we have been talking about cells as individual entities. And then in a previous topic, cell signaling, we saw how cells can communicate with each other. And now I want to stress again that no cell lives in isolation. It is always influenced by neighboring cells and by its neighboring environment. And so, this topic is going to focus on how cells physically interact with one another and how cells physically interact with components in the extracellular space. So, in multicellular organism, cells come together to form tissues, and then these tissues can come together to form a larger functional unit known as an organ. And it is through the cell to cell interaction as well as to cell to matrix interactions that these tissues and organs are maintained. In the extracellular space is an environment known as the extracellular matrix or the ECM. And it contains a concentration of protein and proteoglycans. And so, not only do these cells interact with each other in the context of tissue and organs, but they do also interact with the components of the extracellular matrix. And that is what we are going to discuss in this topic, how cells interact with each other and how cells interact with the components of the extracellular matrix. The way in which cells interact with each other and matrix is through the structure known as junctions. And these are a large collection of proteins with very large structures that are visible by microscopy. Definitely visible by electron microscopy, and they are often visible by fluorescence microscopy, especially if you have the right fluorescence reagent and specific antibodies to view the various components of the junction. And so there are three main groupings of junction. And I think what might surprise you is that we have looked at all of these structures in other contexts throughout the course of this semester. So one type of junction is known as this occluding junction. And an example of an occluding junction is a tight junction. And we looked at tight junctions when we were studying transmembrane proteins and transport cross membranes. The second type, major type of junction, is known as the anchoring junction. And as the name suggests, the role of anchoring junction is to provide structural integrity to the tissues. And anchoring junctions are used by cells to interact with other cells or with the matrix, or for cells to connect to other cells and to the matrix, and they do so by utilizing the components of their cytoskeleton. So there are four types of anchoring junctions. First of all are the anchoring junctions that utilize actin. So when cells interact with each other and utilize actin, the junction is often referred to as adhesion belt. And we saw adhesion belts when we were talking about cellular cytoskeleton and in particular actin. When the cells interact with the matrix and utilize actin at the same time, they form a junction known as focal contacts. And we also saw focal contacts when we were discussing actin. Cells can interact with each other at the same time, utilize the intermediate filaments, and these junctions are known as desmosomes. And cells can interact with the matrix and at the same time utilize intermediate filaments, and these type of junctions are called hemidesmosomes or hemidesmosomes. And these are probably the only two new structures that we will encounter in our discussion on cell-to-cell and cell matrix interactions. And finally, the third type of junctions are known as communicating junctions, and it's through these type of junction that the cells communicate with each other. And an example of communication junction is a gap junctions. And we also introduced the gap junction in cell signaling lectures. So what I want to do is to look at each one of these classification of junctions and look at examples of them. What is interesting is that you can visualize all of these junctions in an epithelial cell layer. And I want to go through these on this figure, but I won't do it in order. At position 6 right here are tight junctions. And often what you see is just below tight junctions are actin-associated adherence junctions, which is shown as number 8. You can often visualize desmosomes, which are intermediate filament-based junctions, as number 9. And at the bottom of this epithelial layer, you can see the cell interacting with the extracellular matrix, which is number 3. Shown in number 10 is a hemidesmosome, which is an intermediate filament-based cell matrix junction. And not shown on the slide are the focal contacts, which would be actin-based cell matrix interactions. And finally, in number 7, you usually see these below adherence junctions are the gap junctions, and these are the communicating junctions. So let's take a look at these different types of junctions and the role they play in the cell. The first major groups are the occluded junctions, and as their name suggests, the role of occluding junctions is to occlude certain proteins and certain solutes. And an example of an occluding junction is a tight junctions, and tight junctions play a number of important roles, especially in epithelial layer. And you have seen a couple of these already. Here is a diagram of the epithelial layer, and you will remember that the epithelial cells are polarized in that they have an apical plasma membrane, an apical domain, and they have this basolateral domain. The apical domain is shown as the top of this layer in the green, and the basolateral layer is the rest of the membrane. And proteins in the apical membrane, such as the protein A, in this example, can freely move within the apical domain, but it cannot pass over to the basolateral domain. Likewise, the basolateral membrane-specific proteins, in this example, protein B, these type of proteins can usually move freely within the basolateral domain, but cannot cross over to the apical domain. And for this reason, the epithelial cells are referred to as polarized because they have a different proteins on one end compared to the other. And it is the tight junctions, as we learned quite a while ago when we were talking about membrane biology, it is the tight junctions that can help maintain that polarity. Tight junctions are structures that are formed near the apical domain of the epithelial cells, producing a very, very tight membrane seal, such as the apical membrane proteins cannot diffuse into the basolateral domain, and the basolateral proteins cannot diffuse into the apical domain. So one of the functions of this tight junction, which is an occluding junction, is to maintain the polarity in epithelial cells. And again, this should be a review. Tight junctions also allow epithelial layers to serve as barriers to the solute diffusion. And so solutes on the apical side of the epithelial sheet cannot pass over to the basolateral side, to the extracellular space of the basolateral side because of this tight junction. And this is really well illustrated in this experiment here. What you can do that you can take some kind of a tracer molecule and put it on one side of the epithelial layer. So for example, in this diagram on the left-hand side, you have a tracer molecule that is maybe electron dense so that you can visualize on an electron microscope. But you can imagine that you can also do this experiment with a fluorescent tracer molecule to monitor the position of the solute. And you can see that on the right-hand side in the figure B, if you take the electron-dense tracer molecule and you put it on the apical side of the epithelial sheet, what you will notice is that you have a strong electron-dense signal on the apical side, but this tracer molecule cannot pass this tight junction over here and flow into the basolateral side. On the other hand, you can take an electron-dense material, electron-dense solute, and put it on the basolateral side, and you can see that it can travel all the way up to the point of the tight junction, but it can never pass through the point of the tight junctions into the apical side of the epithelial sheet. So these tight junctions allow the sheets to serve as barriers of cellular diffusion. And here is another example which shows the use of an electron-dense tracer known as lanthanum hydroxide. And it was placed on the basolateral side of the epithelial sheet. And you can see that the tight junction prevents the flow of that solute past that sealed membrane onto the apical side. Now, of course, there are exceptions. Tight junctions are not completely impermeant to all components, and there is a pathway known as paracellular pathway. Substances, certain small molecules and ions can move from one side of the epithelium to the other through parts of the tight junction, or extracellularly or through the space between the cell. This pathway is in contrast to the transcellular transport, where substances pass through the cell. And there are a handful of components that can pass through the tight junctions. And in fact, we have already seen one of these. Recall our discussion on CFTR, which is that the important chloride channel chloride transporter that is a member of the ABC superfamily of transporters, and an absence of the CFTR or mutation of CFTR is implicated in cystic fibrosis. And you can see in the upper panel a functioning CFTR that is pumping chloride from the intracellular space into the apical side of the epithelium, into the airway lumen. And now you have a net negative charge, which can then attract sodium flow from the basolateral side of the epithelium to the apical side. And so sodium is an example of a solute or a small component that can actually cross the tight junctions. But these are very, very rare. There are not many of these small molecules that can cross through the tight junctions. Another really important role of the tight junction is in the transcellular transport, and again, we saw this when we were studying membrane biology. And you will remember this is an example and the figure showing the transport of glucose from the intestinal lumen, which is on the apical side of the epithelium or the epithelial lining of the intestine, all the way across the cell, and then release of the glucose to the basolateral side of that epithelium. And this is known as the transcellular transport. And transcellular transport relies specifically on two different transporters in the plasma membrane. The first, labeled as number 2, is a sodium glucose symporter. Remember, this is a coupled receptor, and this symporter can move glucose against its concentration gradient while moving two sodium ions down its concentration gradient. So remember, this pump utilizes the energy stored in the sodium gradient to move glucose against its concentration gradient, just like a revolving door. Once glucose is in the cytosol, it can quickly be transported out of the basolateral side through the action of GLUT2, which is labeled as number 3 in this slide. And you remember the GLUT transporters participate in facilitated diffusion and they move glucose down the concentration gradient. Now, the tight junction is very important in this process because imagine what would happen if there was no tight junctions at all in the epithelial layer. You can imagine that the glucose might abnormally flow from the apical side of this epithelium into the basolateral side in between the cells rather than going through the cell. And this can really mess up the glucose gradient to the point where this GLUT transporter might work in the opposite direction and transport glucose from the basolateral side to the intracellular space rather than in the opposite directions. And so the tight junctions really help in maintaining these gradients on the apical side and on the basolateral side, which is important for the function of all these various types of transporter or pumps. This is a diagram of the structure of a tight junction. Tight junctions are made up of three types of proteins, and these proteins come together to form the tight junctions. Occludin and claudin are multi-pass transmembrane proteins. JAM is a single-pass transmembrane protein. And these three proteins can come together to form these very tight bands of proteins that run both horizontally and vertically. And together they form a very, very tight seal, which is a tight junction and is obvious in these electron microscopic image. So let's move on now to the second type of junction known as anchoring junctions. And as their name suggests, anchoring junctions confer structural integrity to tissues. And so it is not surprising that you see a high abundance of anchoring junctions in heart muscle cells and in skin epithelial cell, because these types of tissues are under significant mechanical stress. And anchoring junctions will utilize one of the two cytoskeletal filaments, either actin or intermediate filaments. So when cells interact with each other through actin, the junction is known as an adhesion belt. And that's the first of the actin-based anchoring junctions. When cells interact with the extracellular matrix, and again utilizing actin, these junctions are known as focal contacts. So focal contacts and adhesion belts are examples of actin-based anchoring junctions. And I should mention that actin-based anchoring junctions are sometimes referred to as adherence junctions. When cells interact with each other and utilize intermediate filament on the other hand, the junction is referred to as a desmosome. And when cells interact with the components of the matrix through the intermediate filaments, the junction is referred to as hemidesmosome. And desmosomes and the hemidesmosomes are probably the only new junctions that you will see in this discussion. All the other junctions we have covered in some other context throughout this course. What is interesting is that even though there are 4 different kinds of these anchoring junctions, they all have a very similar general structure, which is illustrated in this slide. So in terms of the cell-to-cell anchoring junction, there is always some type of transmembrane linker protein shown in the green, which interacts with the transmembrane linker protein of the neighboring cell. So all these junctions are transmembrane proteins that interact with the transmembrane proteins on a neighboring cell. On the cytosolic side of these transmembrane linker proteins are a collection of proteins known as the intracellular attachment proteins, shown in the blue. And these proteins that connect the transmembrane linker proteins to whatever cytoskeletal filaments being utilized is shown in the red, which can be either actin or the intermediate filament. All right, the same is true for the cell matrix interaction. You have a transmembrane linker protein shown in green here at the bottom of these cells, which interacts with a component of an extracellular matrix. And then you have these intracellular attachment proteins, which associate with the cytosolic side of the transmembrane linker protein and connect that transmembrane linker protein to a cytoskeletal filament. Sometimes these intracellular attachment proteins are called intracellular attachment plaques because they form a patch of proteins that connect the transmembrane linker protein to the cytoskeletal filament. So let's look at some examples of each of these 4 different types of anchoring junctions. Cell-to-cell anchoring junctions that use actin are known as the adherence junctions. And these adherence junctions can be a part of what is known as adhesion belts, which I'm going to show you in a moment. But here is a transmission EM of an epithelial layer, and you can see the adherence junctions just below the tight junction, which is where often see the adherence junctions just below the tight junctions. So as I mentioned, the adherence junctions are the part of these larger structures known as the adhesion belts. Adhesion belts are very, very wide bands of actin and myosin 2 filaments, and they form a very thick belt just below the tight junctions in the epithelial cell layer. Again, these are called as adhesion belts. And in this case, the transmembrane linker protein is a protein known as E-cadherin. And connecting the cadherin to the actin are a wide variety of intracellular attachment proteins or plaques. Some example of these are catenin and vinculin. And in the second part of this discussion on cellular interactions, we are going to look specifically at these proteins. But for now, these adhesion belts, the transmembrane linker protein is cadherin, and intracellular attachment proteins can be such proteins of catenin and vinculin. So adhesion belts are very, very important because they are anchoring junctions. So their main function is to provide strength and structural integrity to epithelial cell layers. Epithelial cell layers are usually under significant mechanical stress. And so these adhesion belts provide some strength to those layers. But adhesion belts also have a very interesting function in vertebrate development. In vertebrate development, there is the formation of a structure which is known as a neural tube. And neural tubes go on in development to form the brain and spinal cord of vertebrates. So it is very important that the neural tube must be formed properly, and the neural tubes are formed from epithelial cell layers. And in fact, they are formed through the function of these adhesion belts. At the top of the slide, you can see a sheet of epithelial cells, and these adhesion belts are composed of actin and myosin in green. And you remember that the myosin is an actin-based motor protein that can cause the contraction of actin. So what happens is myosin 2 can induce contraction of these adhesion belt shown here in the second step. And this causes a puckering or pinching of the epithelial cell layer at the apex. And eventually this epithelial sheet will roll up and pinch off to form the neural tube, which again will go on to form the brain and the spinal cord. So you can almost think of it as pulling the drawstring on a purse, for example. So it is this adhesion belt that shrinks and causes the puckering or the pinching of the neural tube into the epithelial layer. So when cells interact with the matrix through actin, they form these junctions known as focal contacts. And again, focal contacts are very, very important in motility, as we will see in a minute. And again, this should be a review to you. We talked about focal contacts. Sometimes they are called focal adhesions. And this helps cell to bind to the extracellular matrix. Or if, in the case of tissue culture cells, to artificial substratum, such as a coating on a tissue culture flask. And you will also remember that the cells that are motile usually have a band or a very, very large concentration of actin around the periphery of the cell. And this actin, through focal contacts, can then adhere to the extracellular matrix or to the surface of the tissue culture flask. In the case of focal contacts, the transmembrane linker protein is known as integrin, and the intracellular attachment proteins are usually talin or alpha-actinin. And it is the talin and alpha-actinin that connect the integrin to the actin. And again, when we start into the second part, we will look at these proteins in detail, and we will look at the structure of integrin as well. And it is very important for the cells to be able to regulate their focal contacts or focal adhesion. You will remember this slide, which is a slide that illustrates the movement of a motile cell. And here in the step 1, The cell is bound to either an extracellular matrix or the tissue culture flask, and they can extend its lamellipodium. And then in step 2, that lamellipodium touches down onto the surface and new focal contacts or focal adhesions are made. In step 3, you have the translocation, which is the movement of the cell body, in this case, forward or towards the right. And then you get the de-adhesion at the back of the cell, so that is when the cell is breaking these focal contacts, and it's very interesting that the cells can actually regulate these interactions such as focal adhesion. And again, in the second part of this discussion, we're going to talk about the molecular mechanism of the formation and breaking of these contacts. When cells interact with each other through intermediate filaments, the anchoring junction is known as a desmosome. Again, this is a figure of a transmission EM, and you can see the desmosome that is right below the adherence junctions or the adhesion belt. And this is the usual position for these type of junctions. This is another transmission EM of higher resolution of a desmosome, and you can see very, very clearly the cytoplasmic plaques and the intermediate filaments. This is another drawing of the desmosome to help you understand the structure a little bit better. The transmembrane linker proteins in desmosomes are called desmoglein and desmocollin, and these are actually members of the cadherin family. So remember, cadherin was a transmembrane linker protein in the adhesion belt. So cadherin is also transmembrane linker protein in desmosome because desmoglein and desmocollin are the members of the cadherin family. Then you can see that the intracellular attachment protein in the blue, and examples of the intracellular attachment protein in desmosomes are plakoglobin and desmoplakin. And finally, you can see the collection of plaque proteins interacting with the intermediate filaments. In the case of epithelial cells, these intermediate filaments are probably keratin. In the case of heart muscle cell, these intermediate filaments are probably desmin. Now, there is a genetic disorder known as pemphigus vulgaris. It is an autoimmune disorder in which individuals make antibodies to desmoglein. They make antibodies to their own desmoglein, and these antibodies can then prevent interaction between these transmembrane proteins. And this caused the loss of integrity in the desmosomes. You have the formation of fluid as extracellular matrix flows between these cells abnormally. But what is interesting is that it primarily affects skin desmosomes, or the symptoms appear because of their effect on skin desmosomes. Probably other organs are affected, but to a lesser extent. But because these structures are so important in skin epithelial layer, if you disrupt them, you can have very serious consequences. And here is an image of a serious large blister that has formed on the scalp of an individual who has this disorder. And in the second slide is an example of blisters that can form, and in this case, it's on the back of an individual who has this genetic disorder. When cells interact with the matrix through intermediate filamentous and anchoring junctions is known as hemidesmosomes. In this case, the transmembrane linker protein is integrin, just like for focal contacts, and the intracellular attachment proteins are related to desmoplakin. They are very, very similar for homologous desmoplakin that we just saw in desmosomes, so they are desmoplakin-like proteins. And here you can see the ends of the intermediate filament that are buried into the plaque protein. Now on this slide, there is a summary of 4 anchoring junctions that we just talked about: the adhesion belt, the desmosomes, focal contacts, hemidesmosomes. And across the top are the 4 categories: the name of the cytoskeletal filament that is being used, the transmembrane linker proteins that is utilized, the intracellular attachment protein that is utilized, and what the transmembrane linker protein binds to, so the extracellular ligand. So you should know all four of these details for all of these anchoring junctions. The last major group of junctions are the gap junctions. And again, here is an example of the same transmission EM of an epithelial cell layer. And you can see that there is a gap junction is located right next to the desmosome or just below the desmosome if you consider the apical side as the top layer of the epithelial layer. The gap junctions are very interesting. We looked at these briefly in our cell signaling discussions, and these are channels that form between very closely opposed plasma membrane. And these channels or these pores allow the flow of very small signaling molecule from one cell to the other. So the gap junctions are a way in which the cells also communicate with each other. And you can see in these EM images the structure of gap junction in cells very clearly. So what is the size of these signaling molecules that can pass through a gap junctions? This can be easily determined by using fluorescent tracer molecule of varying size. So in this experiment, what you can do, you can take a variety of fluorescent tracer molecules of different sizes. So for example, 100 Dalton, 1000 Dalton, 5000 Dalton, or 20,000 Daltons. And you can micro-inject them into one cell that is presumably connected to the other cell through the gap junctions. And then using a fluorescence microscopy, you can watch the movement of the fluorescence tracer molecules from the injected cell to the other cell. And if you really did this experiment, what you would find is the fluorescent tracer molecules of about 100 Dalton or even 1000 Dalton will readily flow to the neighboring cell, whereas the fluorescent tracing molecule greater than 1000 Dalton will not cross the gap junction. So signaling molecules such as inorganic ions, amino acids, nucleotides, vitamins can cross into the neighboring cell through the gap junctions. But larger signaling molecules such as proteins, peptides, nucleic acid, polysaccharides would likely never cross the gap junctions. So this is the structure of the gap junction. It is formed by a protein known as connexin. And connexin is a 4-pass transmembrane protein, and 6 of these connexin molecules can come together in the membrane, as shown here in the upper right corner, to form a pore. And this is known as a hemichannel or connexin on one cell. And what's interesting is that the hemichannel is one cell can align with a hemichannel in another cell and also made up of 6 subunits of the connexin molecule. And in doing so, a pore reaches across both the plasma membranes. Another interesting thing is about the gap junction is that they actually can be regulated. So the connexin subunits within the hemichannel can take on different configurations, such as that the gap junctions can be closed or opened. In the cytosol, when you have a high concentration of calcium and very low pH, gap junctions are usually closed. The connexin molecules are such way that they form a structure in which there is no pore. And when calcium levels are very low and the pH is very high, these pores can open. So it's very, very interesting that cells can regulate the permeability of the gap junctions. This concludes part 1 of our discussion on cellular interactions.